this. So we're going to go from there and talk in more detail about what's actually happening at the subcellular level here. Okay, so next slide. Oh, right, I have another video. Okay.
Myofilaments are
okay. So if you remember from Friday, maybe on the actin, there's these two pieces that are called tropomyosin and troponin. They essentially are what is regulating contractions. So if we didn't have tropomyosin and troponin, our muscles would constantly be contracting, which sounds really painful, like one huge muscle cramp all the time. So these tropomyosin and troponin molecules on the actin specifically are regulating and basically not allowing contractions to happen until there is an activation signal sent to the muscle. And we'll talk about the activation signal today. The contraction happens essentially, and I think I mentioned this sort of quickly on Friday, when calcium ions flow into that cell. So calcium ions are what is going to allow for the muscle to actually contract. So what happens, and you'll see this in this next video, calcium ions are binding to troponin. That's going to move the tropomyosin off of the active sites so actin and myosin can actually bind. And I'll show you, I think I have another image of this because that's kind of hard to imagine. But on these actin filaments, you have that troponin and tropomyosin. It needs to actually be moved out of the way in order for the myosin to bind to the actin and to allow for that contraction that we just saw happen. So myosin has to bind to the actin here. That's what's going to allow for contraction. Without calcium, that's not possible. An inflow of calcium is going to allow that to happen, and we'll talk about that in a little bit more detail. This video kind of shows it. The beginning of this video has a little bit of extra information that we haven't covered yet about something called action potential, so an electric wave coming through. We'll talk about that today, but don't worry if you have no idea what they're talking about there. So let's watch this video.
an action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the...
So all of that, don't worry too much about that. We're going to talk about it. It's basically just an electrical signal that's being sent from the nerve into the muscle, telling it to release calcium, which we'll see next.
Sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament.
So that's really important. The tropomyosin is what's blocking the active sites. The active sites are what myosin needs exposed in order to bind to it. So we'll see that those being exposed next.
Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction,
which we just talked about. So we talked about, and I'll mention cross bridge again, but that's why calcium is so important. It has to move, it has to bind to troponin to move the tropomyosin. I know it's like a lot of different individual steps there and individual pieces, but all of those are important, and I'll definitely ask you about them on this exam as well as the final. Okay. Back to it. Oh, wait. Yeah, okay. Share that.
And action.
All right.
And action.
Okay, so into the striations. So what's actually happening here? We know that actin and myosin are binding to each other. And we also know that this has something to do with the striations that we saw in the muscle tissue, the skeletal muscle tissue. It has those stripes, right? So the stripes are essentially one of these sarcomeres. So you have the borders here. So one sarcomere is bordered by what we call Z discs. Sometimes you'll see Z disc, Z line, Z band, Z something. It's obvious why it's called a Z whatever, Z disc, for example. So this is going to delineate a sarcomere. So two of those, everything in between that is considered a sarcomere. That's what actually contracts those individual pieces. And that's kind of one of those stripes. That's a striation within one of those muscle tissues that we saw. So the sarcomere is the contractile unit. There's a bunch of these all along a muscle cell that are all contracting, hopefully at the same time, to allow the overall muscle organ as a whole to contract. So you have the Z discs. Those are really important aspects of the sarcomere. Those will come together when a contraction happens. The other important aspect that you need to know is the H band. The H band is this area here, and this is a relaxed muscle. So the actin filaments are not anywhere near each other. So these orangish things are the actin filaments. When they're separated like that, you have this H band in the middle. That's a resting muscle. When the muscle contracts, that H band essentially disappears. So the actin filaments walk towards each other. So you no longer have that space in between the actin filaments. So this H band area here will disappear upon muscle contraction. So the Z discs come together, the H band disappears. These greenish things here, we didn't really talk about. You don't have to worry too much about them. They're just elastic filaments. The actin filament is actually attached to the Z disc. The myosin filament is not. So here you have the actin filament attached to the Z disc. The myosin filament is kind of in between, attached indirectly. Okay, so Z discs and H band and sarcomere, those are all terms you want to know and be able to identify. And another video. All right. So here we're going to look at an actual sarcomere contraction, kind of see how that happens between the Z discs.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
You don't have to worry about the I band or the A band. I'm not going to require you guys to know those. You just want to know the H zone, which disappears, and then the two Z bands will come a little bit closer together upon contraction.
Yeah.
Okay. Oh yeah, before we get into this, we're gonna do that worksheet that I mentioned. So for you guys on Zoom, you should have found, or if you haven't yet, go to Canvas under the modules, or it's under files as well. If you look for the, wait, what did I call it? The muscular tissue worksheet. It should look something like this. This will be the first page. And then on the back, just a few questions with that figure that I just showed you. So we're gonna work on these for a few minutes, just kind of on your own. I'm not gonna put you in breakout groups, but try to fill in, for you guys, I need to get you the word bank that I have up here in the room somehow. So download that and start kind of at least making note of what these different pieces of the muscle are. I'm going to hand it out. So you guys can work together if you want to here. The word bank for this first page is on the whiteboard over there. So this diagram is, we talked about kind of the makeup of a muscle tissue. So this part, muscles are kind of tubes within tubes within tubes. So being able to label those and identifying what the different tubes are is really important. So that's why I'm having you guys do this. And then the back of it is essentially what we just went over, those sarcomere terms. So work on that and we'll go over it in just a few minutes. Okay. So this concept of tubes within tubes within tubes can be really confusing. So 1 through 4 was probably the toughest for you, trying to figure out what you're looking at. So if we start at the grossest, largest level, we're looking at the biceps brachii muscle, the bicep muscle in the arm. So what would number 1 be of those options? You guys on Zoom can chime in too if you want. What's the overall called? Muscle. Yes, I hope everyone got that. Okay, that is a muscle. So when we're talking about a muscle, we're talking about the organ. It's a whole bunch of muscle cells put together. Okay, so number 2, we're zooming in a little bit. So that would be this, so one of these kind of circles, one of these bundles. What would that be called? One level down from the muscle.
That's
here until someone answers. So there's only 3 other options. Oh, sorry.
Is the fascicle?
Yeah, the fascicle. Yep. So that's the fascicle. So a bunch of fascicles make up the organ, the muscle organ. Okay. So you have fascicle right here, and then within each fascicle, what are there a bunch of? Muscle fibers. Yeah. So muscle fibers or myofibers, those are the individual muscle cells. So here you have, what are we, 3? Yeah. This is going to be a muscle cell. And we still have more tubes within that. So what are the tubes within a muscle cell called? So these little things here. The only one that's left. Myofibril. Yeah, so a myofiber is the cell. Myofibril are the little tubes. They're made up of the actin and myosin within the cell. Okay. The connective tissues here. So these are the tissues that are kind of holding everything together, right? The different mesonems. So the outermost around the entire muscle, which one is that called? Number 5. What's holding? Epimysium? Yeah, so that's the epimysium. Epi means on top of or most superior. So that's going to be the one that's around the entire muscle. So around your entire biceps brachii muscle is the epimysium. Next down, so around one fascicle. What's that one called? Around a fascicle. So holding a bunch of cells together, basically. What's that one? What is the connective tissue that holds fascicles together?
Myofibril.
The myofibril is the, that's what's within a cell. So within one muscle cell. So myofibril was the answer to 4 up here. So who knows what the connective tissue is that wraps around a fascicle? Paramecium, yeah. So the paramecium is wrapping around a fascicle. It's essentially holding a bunch of cells together. And then the last option is endomysium. So that's the connective tissue that surrounds one single cell. So one muscle fiber is going to be surrounded by the endomysium, and endo means inside. Okay, so you definitely want to know those. And then the structure of a sarcomere, we just went over this. So hopefully this is pretty easy. I didn't talk much about the first one, but you guys can probably figure out what it is. So these little globular things, what are those called? What? Myosin heads, yeah. Oh, this is painful, you guys. Myosin heads are on the myosin, right? Those are what bind to the actin. So myosin heads, and then so that makes, what does that make number 9? If myosin heads are there, what does it make this filament? A myosin filament, yep. So there's myosin heads, there's a myosin filament, it's the thick filament. You want to know both of those names, thick as well as myosin. 10, what is this filament called? Actin or thin, yeah, exactly. So that's the actin filament or the thin filament. And then 11, around what's the structure that's on either side of a sarcomere? A Z disc, yeah. So it doesn't look like a Z here, but you know it's one sarcomere because you have those myosins and the actins are attached to it. So that makes it a Z disc or Z band. Okay. I will ask you about those on exams. So you definitely want to know those different pieces, and I will for sure ask you about this in one way or another on exams. So you really need to know the structure of a sarcomere. So this is kind of the finest, one of the finest, almost finest level of detail we're talking about. Okay, so the Z line, well, it represents the Z line here. Yeah, the squiggly things on the end, right? The Z's. So that's those are the Z lines or the Z bands. So right here, you would label that as Z line. The H band, where would you describe the H band as being? Yep, the middle section. So right here, that space between the actins. So this is a relaxed muscle, so it still has its H band. Right here would be the H band. So that whole space, not just the line in the middle. And then what color are the actin filaments? Which are the actin filaments here? Like reddish orange, yeah. So these are the actin filaments. That's gonna make these the myosin. The purple are the myosin filaments. So you have myosin here, actin here, thick and thin, very obvious. Okay, so that makes this entire thing a sarcomere. Everything between the Z discs is a sarcomere. So all of this is one sarcomere. Any questions about that? Like the different pieces of a sarcomere? Okay, I should have reworded this, but we'll say which segment of this myofibril disappears when a muscle contracts. So a couple different pieces shrink. What segment disappears? The H band, yeah. So the H band in the middle completely disappears. So the actin filaments are actually overlapping each other in a contracted muscle. So this section here, the H band will disappear. So the actin filaments are contacting each other in a contracted muscle. The Z discs also kind of come together a little bit. So the whole sarcomere shortens. So when I said which contracts or which section shrinks, that would kind of both of them, the whole sarcomere as well as that H band. The H band is the one that actually disappears. Okay, and then finally, which of these filaments actually moves towards the center during a contraction? The thick or the thin? The thin, yeah, the actin is the one that's moving. The myosin doesn't move. The myosin kind of stays in place and pushes the actin along towards the middle. So it grabs onto the actin and pushes it towards the middle of the sarcomere. So yeah, the actin will move, the myosin will stay in one place. The heads will just grab onto the actin and shift it. Okay. Let's go back to that. Okay, so you guys can hang on to that. You want to study it. I'll be asking questions about it. Probably not the exact same questions, but if you understand those concepts, then you should be able to answer questions about that material. Like I said, this is, I mean, I would say this section that we're doing right now and are getting into is the most complicated material, at least to understand in this unit, the muscle tissue and how muscles actually contract. Okay, so, oh. Sorry, Madison. Yeah, if, yeah, sorry, I didn't see your chat until recently. Sorry if you can't see me pointing on the board. I'm trying to also come back and point on Zoom. I know it's not ideal. I apologize. Okay, so nerve-muscle relationship. Muscles don't contract without a nerve actually sending a signal to the muscle to contract. So we haven't talked about the nervous system. We will get into the nervous system after we finish up the muscular system. So I just need to kind of touch on what we're, how the nerve muscle relationship works. So this will be really brief, kind of an intro to a little bit of nervous system stuff. Okay, so our muscles don't contract unless, like I said, a nerve actually sends a signal to our muscles to contract. These relationships are accomplished by somatic motor neurons. So the neurons, if you remember, are those main cell bodies of nervous tissue that have the branches coming off. So the nerve cells, those neurons, the main body is for these skeletal muscles that we're talking about are found in either the brain stem, so the base of the brain, or in the spinal cord. So we're not talking about anything up in the kind of upper reaches of the brain. It's all spinal cord and brainstem. The axons, which are one of those branches that come off of the main neuron cell body, those are what send messages out from the nerve cell. They all actually reach into individual muscle fibers. So they can be really long. Obviously, if you have a neuron, the cell body in the spinal cord, and then the axon reaching out to, say, the tip of your thumb, it's a really long axon. It has to reach all the way from the spinal cord to the thumb. So these axons are really long, but that's what's making the connection between the central nervous system and the muscles that are contracting. What these neurons are doing when they're telling a muscle to contract is they're telling the muscle to, or sorry, they're releasing acetylcholine, which is a neurotransmitter. You'll see this term a lot. We'll talk about it here in muscles. We'll also talk about it in the nervous system. So ACH, or acetylcholine, is a really important neurotransmitter. All a neurotransmitter is a chemical that sends a signal to something, to another part of the nervous system or to a muscle to tell it to do something. So it's just a chemical signal. Acetylcholine is the only one I think we deal with in muscular tissue, but there are a lot of other neurotransmitters as well. All right. So this relationship is accomplished by what we call motor units. So these motor units are essentially one nerve fiber and all of the muscle fibers that are innervated by it, which just means all of the muscle fibers, the muscle cells that are controlled by this one neuron. So if we look here, we have the spinal cord, which we'll talk about. You have the cell bodies of neurons in the spinal cord. So here you have the spinal cord, kind of a cross section of it, the neuron cell bodies represented by these spheres. And then the axons are reaching all the way down into the muscle. So here, this looks like the femur. So we're reaching down into maybe the quadricep muscle of the leg. And this axon is what's sending that signal basically to the muscle to contract. So we have a couple here. It can be sending multiple messages. We have both small and large motor units. And the small motor units basically control muscles that have a really fine degree of control, like your fingers and your eyes. It can move very specific ways. Those usually have one neuron will essentially control 3 to 6 muscle fibers. So 3 to 6 cells are controlled by one neuron. That is not many. So those are the small motor units, aptly named. The large motor units are more concerned with strength rather than like dexterity. So these motor units can have one neuron controlling up to, say, 1,000 muscle fibers. So rather than controlling 3 to 6 individual muscle cells, they can control 1,000, and that's going to basically provide a lot of power. So the gastrocnemius muscle, which is your calf muscle, so on the back of your shin, has about 1,000 muscle fibers per neuron. We don't really have a whole lot of dexterity with our calf muscle, right? We can't do a lot of the things that we can do with our fingers with our calf muscle. It doesn't have that dexterity or that fine degree of control, but it has a lot of power. So when you push up off of it, you can push the entire weight of your body, basically. So that's the main difference between small and large motor units. And now we get into some aspects of electrophysiology. So electrophysiology is just a study of how electricity basically runs through our body. That's what's going to allow your muscles and your nerves to fire. So it's all based on electrical currents running throughout your body, which I think I've mentioned before. So we're going to talk about that for the next several slides. So when I say electrically excitable cells, that just means they have a difference in voltage across their membranes. So the voltage or electric potential is that difference in electric charge from the inside of the cell to the outside of the cell. So that difference, the outside of the cell is positively charged, the inside of the cell is negatively charged. That's what allows for an electrical impulse to be released. And then that will cause, after many other steps that we'll talk about, a muscle contraction. So this is kind of the first step in a muscle contraction, releasing an electrical signal. Okay, so we have voltage in our cells within a resting muscle cell. So one that's just not contracting. The charge is about -90 millivolts. I know that number doesn't really mean anything to you, but we'll talk about it in relation to nerve cells as well. So nerve cells are also negative inside. So they have a negative charge inside. But just remember that it's a negative charge inside the cell. That is the most important part. Without the negative charge, we don't have any muscle contractions. So we have to have, when a muscle is resting, a negative internal charge in the cell. That's called the resting membrane potential. And it's maintained by the sodium-potassium pump. So if you remember way back when we talked about the good old sodium-potassium pump, it's critically important to a lot of different aspects, including muscle contraction. So I'll talk a little bit more about it. We won't go into too much detail, but I'll talk about the scenario it sets up to allow for contraction in muscles. So here we're looking at a resting cell. It's not contracting. In a resting cell, like I said, the internal charge is negative. So here you have negative inside the cell, positive outside the cell. So you have that charge separation. Inside negative, outside positive. This is the plasma membrane here, the cell membrane, or the sarcolemma. We're talking about a muscle cell. So the sodium and potassium aspect, they're both positively charged, right? So that's not necessarily where we get all of the negative charge from. But in general, there's more sodium ions outside the cell in the extracellular fluid. So you see all these sodium ions, potassium ions, there's more inside the cell. So there is this gradient where you have a lot of sodium ions outside the cell, a lot of potassium ions inside the cell. That's what the sodium potassium pump is setting up. Basically forcing sodium ions out and potassium ions in. And that's partially contributing to the negative charge, but also these anions, there's a lot more anions or negatively charged ions within the cell. So just bringing that charge down. So these A negatives here are just general anions inside the cell. So if you remember back to the sodium potassium pump, 3 sodium ions go out for every 2 potassium ions coming in. So that also helps bring the charge down a little bit. So more positive charges are going out of the cell versus the number coming in. So with each round of the sodium potassium pump, that charge within the cell goes down into a more negative direction. So I don't have time to get into it today. I'll leave the next slide until Wednesday, but essentially the electricity that's released gets released when these gates, there's gates that open on the plasma membrane and sodium goes in, potassium goes out. That releases an electrical signal down the cell membrane. So in the cell membrane, there's gates here that you can see that will allow potassium out, sodium in. When those flow across the membrane, that sends an electrical signal down the membrane. And then we'll get into the next step as to what that actually does for muscle contraction, but that's the first step. So we'll talk about that in detail on Wednesday. And don't forget your videos, your disease videos are due Friday by 11:59 p.m. If you have any questions about that, feel free to ask or email me. But I'll see you guys on Wednesday.